Great, thanks very much. Good afternoon, everyone. My name is Jim Hayes. And in my lab, we study learning and memory. And in particular, we focus on how the brain is able to keep track of time. That is, how we're able to remember the duration and the sequence of events that occur in our lives. We think that the ability to keep track of time is a fundamental building block for cognition, to remember, or to be able to infer cause and effect, to, for example, know that when a seed is planted, weeks or even months later in time, a plant will grow, or to know when it's safe to cross a busy street, all depend upon the brain's ability to keep track of time. Now, I wasn't always on the path to become a neuroscientist. I grew up in Vancouver, Canada, in a family that was very oriented around sport. In fact, as a college student, I moved to a small town in Wisconsin to take an athletic scholarship in track and cross country. And of all things, the event that I specialized in was actually race walking. This is me competing at the Melrose Games at Madison Square Garden in New York. Now, this time I had no idea I would eventually become a neuroscientist studying how the brain keeps track of time. In fact, my undergraduate training was in economics and math. But I now see, in retrospect, it was during this time that I first became obsessed with the concept of time. I was constantly thinking about qualification times for nationals and for world championships. And I often remember even just jotting down split times for the afternoon's workout in the sides of my course notebook. It was also during this period in my life that I now realize in retrospect I really learned how to push myself and how to keep pushing myself in the face of obstacles and failure, which I would argue is probably the most important aspect of becoming a successful scientist. Now, I eventually became interested in neurobiology, and with my math background, I moved to do my PhD at Boston University with Dr. Michael Hasselmo, where I began using computational methods to model neurophysiology. And I was eventually trained as well as an experimental electrophysiologist. Now, one of the really exciting things about doing neuroscience research today is that we have access to a range of recently developed tools that allow us to ask questions about the brain that would have been essentially science fiction only maybe a decade or so before. And as a postdoctoral fellow, I moved to Northwestern University to train with Dr. Daniel Dombeck. And Dan himself was trained as a physicist in optics and biophysics. And I moved to his lab to help develop some tools that would literally allow us to watch the activity of thousands of individual brain cells in the intact brain in behaving mice as they were learning and performing memory-guided timing behavior. Now, as my postdoctoral fellowship at Northwestern was coming to a close, and I went out onto the faculty job market, I was faced with a really exciting but also a daunting question of where I wanted to start my own lab. And when I discovered the neuroscience community here at the University of Utah, I knew that this was going to be the place that would provide a launching pad for my own lab. I found that there is a culture of collegiality, and there's also a culture of support to go after big ideas. And in fact, through this support to tackle big problems, my lab was recently awarded an NIH New Innovator Award that is specifically designed to fund high risk but high reward research. And to give you more insight into some of the work that we're doing as a part of this grant, in the remaining few minutes, I'm going to tell you a short story about some cutting edge optical tools that we've developed and applied to understand how the brain keeps track of time on the scale of seconds to minutes. So in order to do this, we've developed a novel surgical and optical approach whereby we are able to implant a microprism into the brain of mice. And we're able to ultimately do this to record the activity or the spiking of huge populations of individual neurons. Here we're using a prism, much like you might use on the optical table to direct the light of a microscope. However, here we figured out how to do this through this surgery to chronically implant it into the brain, and that then allowed us to use a powerful type of microscopy called two-photon microscopy and leverage recent developments in genetically encoded optical calcium sensors in order to read out the activity in a part of the brain called entorhinal cortex, which is interestingly the first part of the brain to show pathological signs of Alzheimer's disease. As you can appreciate, it's located deep in the brain in a very hard to reach location. And this was another one of the real motivating factors in using this optical approach. So here you can see some of the raw data that we collect. So what you're looking at is individual neurons flashing. This is because they're emitting tiny electrical pulses or they're spiking. And through this series of approaches, we're able to read this out optically. So the second component to our approach is a novel behavioral approach that leverages virtual reality. So here the mice are positioned underneath our two-photon microscope, which you can see in the schematic. And we're then able to keep the brain relatively still, enabling the use of optical methods. But we can still engage the animal in navigation behavior in VR, much like they might do in more traditional rodent maze tasks. So the way that, in order to understand how the brain keeps track of time, we've developed a virtual navigation task in which mice learn to run down and stop and wait at an invisible door for a given length of time. So here you see the mouse running down. It knows the door is at the interface between these two textures, and it stops here. And we're asking it to wait at this invisible door, in this case for 6 seconds, after which time the door will open. The mouse knows this because it's using its internal sense of time to solve this task, and it reports it to us through this behavior. So we can combine this behavioral approach with our powerful imaging approach in order to record the activity of neurons in entorhinal cortex, and we wanted to know whether or not we could see the signatures of a neural clock in the dynamics of these brain cells. And here's what we've discovered. Here you see that individual neurons become sequentially active when the animal stopped waiting at the door performing the timing task. It's like a clock going tick, 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 tick. And this was part of an exciting series of discoveries that we published in the journal Nature Neuroscience in 2018, suggesting the neural dynamics that underlie the brain's ability to keep track of time. But what you would, I think, really like to do in order to further test this hypothesis is you'd like to go into this clock and manipulate it and then cause changes in the animal's timing behavior. And that's exactly what we've done in a follow-up series of experiments. Now in this case, rather than using optical tools to read out neural activity, we've used a different set of tools, so-called optogenetics, to write or manipulate neural activity. So here we use a viral vector-based approach so that neurons in entorhinal cortex make a light-sensitive protein that when activated with light will block these neurons from firing. And that's what you can see here. So we began using this method, delivering light every time the animals were stopped waiting at the door, trying to time and solve this task. And we did this in control mice with no optogenetic protein made. And what you see is that over sessions, they learn how to solve the task. That is, they tend to wait for the appropriate amount of time. In this case, it's 4 seconds. However, in our experimental group with the mice with this optogenetic protein made, they don't show this same learning. You don't see any peak in this wait time distribution, or in other words, these mice just behave like naive mice like they did on day one. And so by blocking these neural dynamics that look like a clock, we can actually disrupt the animal's ability to tell time. So I've recently started my lab here at the University of Utah. We're continuing this work as part of our NIH New Innovator Award to understand the fine details of exactly how this neural clock works, how these temporal signals are integrated into memory circuits in order to remember the duration of events. I would really like to thank my past and present mentors and members of the Hayes Lab. And I'd really like also to thank my colleagues here at the University of Utah for just the tremendous support that I've received in getting my lab up and running. And with that, I will end and thank you all for your attention.